So hello and welcome again, Microbehunter here. Welcome to another Microscopy podcast, and I would like to answer 5 questions today. Questions that I received from the community all across the world of microbiology and, of course, microscopy. Yeah, some of the questions are a little bit specific, and I want to give you a little bit of background information, of course, as well, to give you a little bit of context. And the questions I received over my YouTube channel, so sometimes people are posting questions beneath some of the videos. Some of the questions are even a little bit off topic, but that's perfectly fine. And some of the questions I also received over the chat of my weekly live stream. And of course, I would like to also invite you to that. Every Saturday, I have a live stream where I'm simply doing a little bit of lab work and showing you some microscope slides and so on. So I would like to quickly read out the 5 questions to you first, and then I'm going to take them one by one. And in the description, you can also see, of course, a timestamp. So the first question is it good to sterilize the equipment in a pressure cooker as an alternative for an autoclave? This is, deep into microbiology methodology. I'm going to, explain everything here. Next question, how do you clean and reuse your cover glasses without cutting your fingers? I throw them out because I always end up cutting myself while trying to clean them. A third question, what are those other tiny organisms swimming around it? And this is to reference to one of my videos that I made. And how many bacteria are not in the database because they keep evolving and new species are constantly emerging? It's also a bacterial identification question. And then last but not least, how do you even identify bacteria? This last question was one of the questions that I got over in my live stream once. So, and because many of these questions are rather specific, I think I'm just going to take a little bit of time to also explain to you the background information to give you a little bit more, more info and why these questions are relevant. So let's go to the first question here. Sir, is it good to sterilize the equipment in a pressure cooker as an alternative for an autoclave? So what's going on here? Why even sterilize equipment, which equipment, and so on? And what are we even talking about here? Well, the question refers to one of the videos that I made where I put a fungus from a rotting orange under the microscope, and I've grown the fungus also in a petri dish. So I cultivated the fungus deliberately so that I've got an easy method to obtain a sample for microscopy, because under the microscope I wanted to observe the spores of the fungus and also how they start to grow. So this is kind of the context here. And every time when you are in a microbiology lab, you have to make sure that any microorganisms that have come in contact with your equipment, petri dishes and whatnot, you have to be killed before you actually throw out all of the equipment. So even if you're using disposable plastic petri dishes and equipment, it's important that you sterilize everything before throwing it out, because you do not want to spread microorganisms around, which might become harmful when inhaled and so on. And you do that by essentially putting the equipment into a pressure cooker. And this pressure cooker is an autoclave, or is called an autoclave. And basically what happens is the following. And to first answer the question, we just use a regular pressure cooker as well? And the answer is yes. As a matter of fact, this is what I've used myself during the laboratory time when I was still at university. We bought a fairly large pressure cooker, and we used that essentially to autoclave and to sterilize equipment. So basically, of course it works, because what is important is that you have a sufficiently high temperature for sufficiently high time. What's going on here and why is this necessary? When you have a pressure cooker, what you do is you put your equipment in there, or maybe even the things that you want to throw away later on, and you add a little bit of water. And then you heat it up and you bring it to a boil, and there is a lid on top of the pressure cooker, and this lid is airtight. And as the water starts to evaporate, what will happen is that the steam pressure will start to build up inside of the pressure cooker, And of course, there is a valve as well to let off any excess steam. Otherwise, there's of course the danger that the pressure cooker explodes. And as the temperature goes up and as the pressure also increases, this allows you to go up to a temperature which is far higher than 100 degrees centigrade. So water boils at 100 degrees, and even if you turn up the heat yet further, the temperature is not going to go up. Right? It's going to stay at 100 degrees. The water is simply going to evaporate faster. Sure, as you go up with the heat, the water evaporates faster, but the temperature is not going to go up. Right? This has to do something with the latent heat of that the water has, and that essentially 100 degrees is not enough to completely sterilize certain microorganisms, because some microorganisms are able to form so-called endospores or spores, and these spores can be highly temperature resistant. So if you want to really make sure that all of the bacteria and their spores are killed off, you have to go up to, let's say, 120 degrees centigrade, maybe for approximately half an hour, and then you can be fairly sure that pretty much everything's been killed off. Right, but 100 degrees alone is not going to kill off everything, and we know that this can be a problem, because back in the day, maybe 100 years ago, 80 to 100 years ago, people still used to make their own canned food. So they made their own tin cans, and they put, let's say, meat and vegetables and so on into those tin cans, and then if it was not boiled hot enough, then there was really a danger of the spores of Clostridium botulinum. That's a very severe form of food poisoning bacterium. The spores of those could actually survive regular boiling water. Right, and so simply boiling the tin cans, in regular water sometimes was not enough, right? And you had to actually go up with the temperature yet further to really make sure that all of those spores are killed off. And this is the reason why, proper, sterile technique is so important. And after you've grown your microorganisms on Petri dishes, therefore you have to, before throwing them out, you also have to autoclave them. Yeah, and to go back to the question here, is an autoclave, that's one of those, is basically nothing more than a large pressure cooker which allows you to sterilize the equipment by heating it up to approximately 120 degrees. And of course, inside you have a lot of pressure, and therefore this autoclave or this pressure cooker must have also emergency valves to let off steam so that the whole thing doesn't explode, right? Because steam explosions can be quite dangerous. For this reason, those pressure cookers that are also used for cooking food, they always contain valves and also emergency valves that allow steam to get off in case pressure is too high. Yeah, I'm digressing again. Just wanted to clarify a little bit the importance of these pressure cookers or autoclaves. So we move on to the next question here, and the next question has to do a little bit with practical microscopy, and is: how do you clean and reuse your cover glasses without cutting your fingers? I throw them out because I always end up cutting myself while trying to clean them. Well, first of all, in microscopy, you have those microscope slides. They're made of fairly solid glass. I generally always rinse them in water and clean them, but there are also cover glasses, and those cover glasses are pretty thin. A standard cover glass thickness is approximately 0.15 to 0.17 millimeters. So they're pretty thin, and they're also, because the glass is so thin, they're also a little bit flexible. You can carefully bend them even a little bit, but of course they're also fragile, and they tend to break easily. So especially if you try to wash them and then you try to rub them between your fingers to remove any dry specimen that might still be on it, there is of course always the danger of cutting yourself. And here, this is where things become interesting. You got to be a little bit careful, because sometimes the material can be infectious, especially if you've got bacterial samples on there, or maybe it doesn't even have to be bacteria, it can be anything else. But if it's not dry and if it decomposes, then you have bacterial growth there, and then there is of course the danger that you cut yourself and that you injure yourself. And so it's completely understandable that you basically don't want to take that risk, and you simply want to throw it out. How do I do that? Well, first of all, I take very much care that I do not have any infectious material on my slides. And if I do have infectious material on the slides, then I'm not even going to reuse them. I'm just going to throw them out. Yeah. For example, if there are certain bacteria that I would like to observe, and then you generally should be careful. And then I simply take the slide plus cover glass as it is, and I kind of throw it away directly. But in most cases, actually in almost all cases, the specimens that I'm observing are really completely harmless. I don't know, cross-section of plant leaves, for example. Yeah, there's really nothing dangerous about those, right? And for this reason, it really depends a lot on what you're observing and how those slides and cover glasses, how they have become contaminated. Yeah, but even if there is nothing dangerous on there, of course, there's always the risk that you cut yourself and injure yourself, which you don't want to do. So what I've been trying to do as well and experimenting around as well is well, maybe you don't, when you wash them, you don't rub them between your fingers to remove the dirt, but you simply allow a detergent to do its activity. So what I do is I take the slides with the cover glasses. Sometimes most cases they're dry and the cover glass sticks to the microscope slide. Yeah, and I simply soak everything in hot water for several minutes. The hot water will, of course, also kill a lot of microorganisms that might be still on there, but it will also kind of dissolve everything that's causes the cover glass to stick to the microscope slide. And in most cases, the microscope cover glass and the slide, they will separate completely on their own. I then add a little bit of dishwashing powder to the cover glasses and the microscope slides with hot water, and I simply let it stand for some time, and then I generously rinse everything. And so basically I allow the dishwashing powder to remove everything that's sticking to the microscope slide. And then there is also no need really to rub the cover glasses or the slides between your fingers to remove any dirt, because this has been done by the dishwashing powder, right? And so for this reason, I generally don't have to take the risk of injuring myself, because I avoided it right from the beginning, from the outset. One of the biggest disadvantage of individually washing the slides and the cover glasses is, of course, that it takes quite a bit of time. You have to wash them one by one, and I therefore also find it easier simply to allow the dishwashing powder to do its work, and then you generously have to rinse everything with clear water at the end. Yeah, generously, because you do not want to have any detergent left over, because even small amounts are able to harm the microorganisms that you want to observe moving around under your microscope. So that's kind of the short answer. How do you clean and reuse your cover glasses without cutting your fingers? Well, I try to handle them as little as possible. Let's put it this way. And as I just mentioned, the dishwashing powder should do its job, and and then you just generally rinse them quite well. So the next question is now the following: what are these other tiny organisms swimming around it? And this question refers to one of the videos that I made, where I think it was the video about Stentor, which is also known as the so-called the untrumpet animalcule. It's not an animal, it's a protist, side issue, right? But in this sample, there was not only the stentor, but there were also many other microorganisms floating around. And the question is now is what are they actually? Well, the short answer is I can actually only answer this question in a very general way, because I don't know what they are. Or do I? Well, it depends really on the level of depth that you would like to identify these organisms. I can tell you already, those other things that were swimming around were also protozoans or protists. It's a very general category, right? Maybe you're still able to distinguish them between ciliates and flagellates, maybe, or amoeba, right? But generally, to go any further in detail, you'd have to really dive in much more. So when you see something moving under the microscope, it's fairly easy in many cases to obtain a general category. But sometimes if you want to go down maybe to the genus level or even the species level, things can very quickly become very advanced. And yeah, in many cases, microscopy alone is also not able to provide enough information, visual information, for you to do a detailed identification. So I can tell you right now, I've also noticed those tiny organisms swimming around, and because they were not very tiny, some of them were also green, so there could be some kind of maybe algae even, and that's pretty much it. I can also tell you that many of the things that we've seen there are not bacteria. There are, of course, bacteria present as well in the sample, but those that you've seen there were not bacteria because I used a very low magnification, and bacteria generally are significantly smaller, and you need a higher magnification to properly see them, right? So you can already see that just by the overall size, I can already narrow down a little bit into which direction it's going, right? But it's not possible for me to really identify a microorganism all the way down to the species level in most cases just by looking at it. It's not going to work. So I can tell you also that in many cases, I don't know what the organisms are, also because maybe some of them are not even in the identification book that I have. And maybe some of them indeed are in the identification book, but they look kind of similar or maybe identical, but maybe they're different species still. Again, because similarity under the microscope does not automatically or in all cases imply that the organisms are also related. Just because two cars have the same color does not mean that they're the same brand of car, to use a very simplistic analogy here, right? So this is basically the thing here. The other tiny organisms swimming around, I can tell you these were probably also protozoans because simply of the size and the general appearance that they have. I'm going to move on yet further, because the last question or the next to the last question are quite interesting here. The next one is also a question about bacterial identification. How many bacteria are not in the database because they keep evolving and new species are constantly emerging? This is a very, very interesting question. And this refers to the following fact that when I explained something once in one of my videos, I also mentioned the importance of DNA sequencing in bacterial identification. This is actually something that I have done myself as well when I was still at university. I had several bacterial strains isolated from the environment, and it was my task now to identify and figure out what they are. And one way of doing that is to determine the base sequence of a DNA, of a section of the DNA, and then check with a database. And then based on percentage similarity, DNA similarity, it will actually tell you what the bacterium could be. And then you're kind of hoping actually that you do not find it in the database, because then this means that you've discovered something new, right? If it's already in the database, unfortunately, somebody else was faster, and the DNA sequence is already in the database. And if it's not in the database yet, or if there's a certain critical percentage deviating, sufficiently different from those in the database, then you can say you've got something new, right? So, however, there are a couple of assumptions here, and in your questionnaire, how many bacteria are not in the database because they keep evolving? Well, most bacteria in the environment are not in the database because only a very small fraction of bacteria have already been characterized and have been sequenced. And in my previous one of my previous videos or podcasts, a couple of episodes back, I actually did talk about that, the reasons why most bacteria cannot be cultured in the laboratory. So the majority of them are not in the database because it's simply not possible for you to culture them, so to determine the DNA sequence. So that's already one thing. However, maybe not a huge problem, because those that are in the database, at least you can then compare your sequence to the existing ones and calculate a percentage similarity. So it doesn't have to be exactly in the database. That's essentially what I want to say. If it's close enough, then you can still determine a sequence, a similarity here, right? And then you're mentioning here is they're not in the database because they keep evolving. That is correct. Bacteria do keep evolving, but I'm claiming right now that evolution is not the primary driving factor why they're not in the database. I think they're simply not in the database because most of them have not yet been characterized. So that's, and how many bacteria there are is their estimates. the number of bacterial species is astronomical because only a small fraction of those have already been characterized. Yeah? And so it's not necessary because of evolution or that they keep on changing. I think the problem is already much earlier than that most of them have not even been characterized yet. And new species keep constantly emerging, as I just mentioned. But here, this begs another question: what even is a bacterial species? I mean, if we don't get me started on that topic, because that is an immensely difficult, actually even a philosophical question, because a species definition in bacteria is different than for the higher plants and animals and fungi, for the higher organisms. Let's put it that way, because you consider an animal to be of the same species if they're able to reproduce and they are able to produce fertile offspring. So basically, a species is able to self-maintain itself into the future by reproduction, right? And I want to give you an example here. A horse and a donkey, for example, are two different species. They can reproduce with each other. You can cross a horse and a donkey, and what you get is a mule. Mules are used in some countries for carrying loads, and they're very good also in the mountainous region because they have a very good sense of balance, and apparently mules are also very friendly animals. So crossing a horse and a donkey is possible, but, and it's important, mules are sterile. This means you cannot take two mules and cross them again to have children. It doesn't work, right? Mules are not able to reproduce to have children, offspring. And for this reason, horse and donkey are considered to be separate species. And this works with those animals, but bacteria, how do the bacteria reproduce? Bacteria reproduce by cell division. And this means this form of sexual reproduction that we have in the higher animals that we don't have in bacteria. So we cannot use the same species definition for bacteria as we can use for the higher organisms, right? So it doesn't work there. So you have to define different characteristics. What is a bacterial species? Now you can say, well, what about if bacteria are able to exchange DNA with each other? And they are able to do that. It's called horizontal gene transfer. So can we not use that as a criterion maybe to define bacterial species? But even that doesn't work, unfortunately, because scientists found out that this horizontal gene transfer, the passing of DNA from one bacterium to the next one, can occur over also even very unrelated bacteria. So basically even bacteria that are very far apart. So we cannot count that as a criterion for a species as well. So what you have to do is you have to essentially, if you want to define a new bacterial species, you have to make your case, so to say, by characterizing it and by falling back on some DNA data to make your case. But in any case, you cannot use the traditional definition of a species for those single-celled microbes. So I just wanted to clarify this, simply to illustrate a little bit how complicated this topic is and how complex the question of a bacterial species actually is. Yeah, with protozoans, it's again a little different because they have those so-called the mating types, and they're able to conjugate with each other. So there is a form of sexual reproduction there happening, where you can use this as maybe as a species for a species definition. But for bacteria, this is getting really complicated, right? I know I'm deviating now a little bit from your original question, but essentially I just wanted to say that the database or lack of species, bacterial species in the database, is a very fundamental issue, and the issue even goes philosophical because we still have to decide or at least agree on a common definition of a bacterial species. Let's put it that way. I mean, there are certain agreements already, but as you can see, it's a little bit more complex, the issue. So I think I don't know. Maybe this question is, I think, very interesting, and I think I could talk about this for much longer. Last question now. And equally interesting is this was actually also asked in one of my recent live streams. How do you even identify bacteria? And this is a very interesting question. There's not one way. The gold standard I already mentioned is the sequencing. DNA sequencing, is the so-called the 16 srDNA sequence, has been defined as an international standard to determine bacterial sequence. But there are other possibilities as well. And traditionally, way back in the day, what they've done is they have done some certain growth tests. And in many cases, the bacteria had to be identified because they had medical relevancy. So if you were ill because of, I don't know, an infection, yeah, essentially there are not that many bacteria that you can choose from. There are only comparatively few. I'm not saying few, but I'm saying comparatively few bacteria around that are medically relevant. And for those, you could establish certain growth tests, or you could narrow down what bacteria they were based on simply observing the colony on a petri dish, on how they look like, on whether they were able to break down, let's say, blood agar. Are the cells able, the bacteria able to produce an enzyme that breaks apart the red blood cells, for example? So you could actually do some simple growth tests or is the bacterium even able to grow in a certain medium? So, and then you could actually narrow it down, basically, and then you kind of more or less knew what you already had, what you were infected with. This was the traditional way. And kind of worked with certain medically relevant bacteria. There were standardized methods on how to do that. But you kind of already had only a handful of bacteria to deal with. But the situation becomes way more complex when you're dealing, for example, in research with bacteria that are unknown. Right, then how do you actually identify those? Yeah, what they have done, therefore, is they've a second possibility would be, for example, to test whether certain bacteria are able to metabolize certain substances. Even these methods are also used still in medical testing these days. You basically get yourself a bacterium into pure culture, and then you are testing what enzymes does it have by checking which substances and types of foods is it able to use. And basically you compare that then with a database again, and then you can also again narrow down what the bacterium possibly could be within a certain statistical variation. So basically the first one is DNA sequencing, as I mentioned, and also how do the colonies look like and in what growth medium, and then also now the end products being produced and the ability to metabolize certain nutrients can also be seen as a method that's also used nowadays in the medical field, where the number of bacteria that you have is not that large. So, but then we move on a little bit, and that's essentially then the area that I've been working in at the university is you can try to analyze what the bacterial cell is made of. So that's essentially analytical chemistry. So I had to, for example, grow bacteria in pure culture, and do a variety of analytical biochemical tests on those bacteria. So, for example, the thing that I've been working on a lot is I analyzed the fatty acids of the phospholipids of the cell membrane of the bacteria. Okay, bacteria, just like all cells, have a cell membrane, and this cell membrane is made of phospholipids, and I basically chemically analyzed what these phospholipids are. So I used a lot of gas chromatography to analyze those, right? So and there are many other things that you can analyze, right? The protein pattern, you can analyze the presence of certain substances, whatever, right? Essentially, this is what I've done. I've basically tried to analyze what are the bacteria made of, and then because there was not one central database, I had to dig through thousands of pages of publications, and kind of scratch everything together to kind of narrow down a little bit of what bacterium I actually have. Yeah, and that would be essentially yet another method to identify bacteria is to analyze the composition. What is it made of, the bacteria? Yeah, and then there are are also some modern techniques that are used also in clinical laboratories, especially when you have to quickly identify the bacterium, so-called mass spectrometry. I cannot tell you a lot about this. It seems to be a fairly new method. You're using somehow lasers to vaporize the bacterial cells, and then you're analyzing the parts using a mass spectrometry, and then you're also apparently able to identify what bacteria you have. So you see there are a whole range of different possibilities here, but it all kind of depends on a little bit on actually already knowing what you've got, and then you have to have either database or publications, and so that you can cross-reference your own strains that you've isolated with those that have already been described. Yeah. At the very end, I want to tell you a little bit, somewhat of a funny story. I don't know if it's funny. It was actually for me a little bit awkward, but now many years later, I can laugh about that. When I finished my diploma thesis analyzing and characterizing my bacteria, I did basically pretty much bacteriology. So it's called chemotaxonomy. This was basically the field where I did my thesis. So I did basically the analytical chemistry part. I analyzed them, and I was able to identify them finally after a lot of work. And then at the end, to verify the results, I did a DNA sequencing. And I remember this institute presentation at the very end of my thesis, hundreds of professors and students sitting in the lecture hall, and I presented for a couple of minutes the results of my research, of 1.5 years of research for my thesis. And then at the very end, I kind of presented how I arrived at the conclusion. And at the very end, I presented the DNA sequencing data. And then, it matches. And I was able to identify the bacterium correctly based on the biochemical analysis. And then I remember this one professor actually then all of a sudden raised his hand and said, well, if you could do DNA sequencing right from the beginning, why did you even bother to do all of the analytical chemistry part? Just do the DNA sequencing, then you have the result. Yeah, and I was totally speechless because, of course, he was right. Luckily, my supervisor jumped in and defended me here. And he said something on the order that it's important if you want to describe a new bacterium, it's not simply enough to simply describe it based on DNA sequencing data, but you also have to describe all of the biochemical composition of the bacteria. And this is basically how he kind of saved me. But actually, it's correct. There is, for identification, all you have to do is you have to sequence the DNA, and then you've got the result within a fairly short time. Right, it's the gold standard, so to say. But for me, this was, of course, a little bit of a shocker because this is wow. I spent 1.5 years essentially doing lab work for nothing. All of the work. It wasn't for nothing. I learned a lot. But simply to give you an analogy here, it's almost like, again, I'm going to use my car analogy. You can identify, if you want to, the brand of a car and the type of a car by analyzing the screws that it's made of and all of the different parts and the wiring and the cables and everything. Yeah, and then because there is not a central database, what you do is you end up digging through, I don't know, thousands of pages of manuals and technical documents, and then you kind of figure out at the very end, I finally figured out, based on the size of the screws and the way it's connected, it's a BMW. Right, and you're happy that you finally figured it out. Yeah, and then essentially says, yeah, why didn't you just check the logo of the car? Yeah, and then you already know it's a BMW, right? So what's the whole point of going into all of this analysis part? You would just check the logo and then you'll know it anyway. So what's the point? Yeah, so that's a little bit the way that I felt at the end. But as I just mentioned, I did not feel that the whole thing was wasted because I simply learned incredibly much by this whole process. So what I'm going to do now is I think half an hour has now gone quite quickly. I hope that this podcast again was interesting for you. I do of course want to invite you to subscribe to this channel or to this podcast as well. As I already mentioned, there's a weekly live stream as well. I think I'm going to leave it at that for today. There is also a comment section. Please also leave your comments behind. Happy microbe hunting as always, and see you in the next episode. Bye-bye.